Recording has started.
Okay, excellent.
Maybe I can share my screen in this case.
We like to keep them guessing, Lydia. Not that it is ended. There you go. Okay. Well, thank you everybody for joining us at our first plenary session. I'd like to introduce our first speaker, Professor Lidia Morawska. And you can see her title there, Just Ventilate the Buildings. It's as simple as this, with a question mark. So I can't wait to hear what you have to say about that. So Lydia is a professor at the Queensland University of Technology in Brisbane and the director of the International Laboratory for Air Quality and Health at the same organization. It's a collaborating center with the World Health Organization on research training in the field of air quality and health. Professor Moraska also holds positions as adjunct professor at the Institute for Environmental and Clinical Research at Jinyang University in China, is a vice chancellor fellow for the Global Center for Clean Air Research. In the UK, she's also co-director of the Australia-China Center for Air Quality Science and Management. She conducts fundamental and applied research in the interdisciplinary field of air quality and its impact on human health and the environment, with a specific focus on the science of airborne particulate matter. Professor Moraska is a physicist and received her doctorate in I can't pronounce the university name properly. Thank you. University in Krakow, Poland, for research on radon. She's an author of over 800 journal articles, book chapters, and refereed conference papers. She's been involved at the executive level with a number of relevant national and international professional bodies, and is a member of the Australian Academy of Science and a recipient of numerous scientific awards. So we're very excited, Lydia, for you to join us today. And with that, we'd like to hear your talk today. Thank you.
Many thanks for introducing me. And now I can share my screen.
Just a quick note for the attendees, if you have questions, please type them into the Q&A panel so that we can propose them after the slideshow.
Okay, thanks again for introducing me, for inviting me to this presentation. I'm assuming that you see my screen now.
Yes, we do.
Yes, great. That's always very reassuring. Well, I was very much hoping that this will be the first conference, easier conference, when we'll see each other in person. This is not the case. Well, I hope this is the last that we can't see each other in person. Hopefully indoor air in Finland, that will be the conference. Well, just ventilate the buildings. Is it as simple as this? And I expect that you suspect what the answer would be. I'll be talking about a few aspects: open the window, mechanical ventilation design versus performance, limits of ventilation, disinfecting the air, enforceable indoor air quality standards, and the vision for the future. Is it a new vision, or in fact, is it an old vision? Well, where we are right now, public health authorities and their views and how they have changed. We went a long way from this tweet, well, almost 2 years ago in March, the WHO claiming that the virus is not airborne. Well, it is airborne. Well, to be precise, in the latest brief of the WHO, it is said it is cold airborne. But with it being airborne, it means that it needs to be removed from the air, and how to do it by ventilation. And WHO has done quite a lot in terms of promoting ventilation with this document, Roadmap to Ventilation, published last year. So we'll talk about this. I'll focus for a moment about this, the aspect of ventilation in terms of what to do with the virus, but I'll also talk about other aspects. If you cannot ventilate, do something else. But let's stay with ventilation. So this is the very latest, end of December, recommendation, well, pictorial recommendation by the WHO about good ventilation and how it protects you from COVID. So if you look at these images, what you can see that the focus is on window. Open the window, what to do with the window, cross ventilation. Well, you can put a fan in front of the window, oriented in the right direction. So this is the message, the window and the role of window in ventilation. Now, of course, there have been many public health authorities already doing something and promoting the aspect of ventilation. This is one of the earliest last year, the Dutch Peccia also recommendation what to do. So if you look at this last panel here, Lucht, light, provide plenty of fresh air, open window, let the buildings air well. This message is very common. Just 2 days ago, the Queensland Department of Education issued very similar, finally, recommendation. Open the window, make sure that the air conditioner works well. I'll come back to this, and orient the fan if you have in the right direction. Well, so this is the message from the public health authorities, open the window. Well, hardly a novel concept in terms of ventilation. We've heard about this, and particularly in this community, I don't need to convince anybody about this, but the world has heard about this. So this is Florence Nightingale, and this publication 1859. So we are only 160 years behind, 163 to be precise, in the context, in the concept of promoting ventilation. Now, what does it mean in practice, this aspect, open the window? I could give many examples of this, what I've read, what I've seen, what I've experienced. Just this one, a UK university, which I know very well and I don't want to name it, autumn 2021. Well, open the window, or windows open to lower the risk of ventilation, but it is cold, maybe not that cold that snow is falling, but sufficiently cold. Heaters, heater on, heater left overnight, caught fire. Well, then there is a ban on self-purchase heaters, but the window is open and heaters are still on. Slightly different situation in Brisbane. We don't have snowflakes in the air, but it's winter, it is cold. So windows open, but because it's called air conditioner on, set on heating. Remember, I mentioned the Department of Education recommending maintenance of air conditioners. And in this case, CO2 concentration measured and kept just above outdoor air. So that's great. Open window ventilation is right, but of course, huge energy waste. This is not for the direction for the society to go. So let's stay for the moment with this aspect of natural ventilation. This natural ventilation in homes, in schools, in restaurants, in shops, and many, many other venues. So when it's not too cold, not too hot, not too noisy, of course, windows are open and perhaps ventilation is right. But the reality is that in most climates, most of the time, it is either too cold, too hot, too noisy, or too polluted outside. So windows are closed and there is no ventilation. So in the reality, natural ventilation means no ventilation. And it is like saying from this children tale, the emperor's new clothes, the king is naked, there's no ventilation. Now, of course, I'd like to make here a disclaimer that when I say no ventilation means minimal ventilation. Of course, the building envelopes always leaks, so there's some air coming. And some countries, jurisdictions, and so on are either more or less sophisticated in regarding the rules for ventilation. So there is always some air coming, of course. Well, can natural ventilation actually mean ventilation? There have been studies published on how to work with in this situation, and I like this one published by Arsen Melikov and colleagues. What to do, and this was the example on schools, this intermittent ventilation. So in this case, the time particle concentration infected person is inside, well, so assuming that one child is infected or teacher, infected person inside all the time, or if infected person is inside and outside, which means if the classrooms, if the lessons have breaks. So this is what happens. So yes, it improves the situation, but in reality, how many schools of departments of education will really want to flexibly change school schedules to adjust to this situation of ventilation? Very few. So again, there's no ventilation. Well, how to improve indoor air quality without ventilation? Since the starting point here is in the context of the pandemic, which is focused on the infection transmission, so let's use filter-based cleaners, and that's what I'm stressing. Air cleaners using filters like HEPA filters. Will it work? There have been many studies published during the pandemic and before, in fact, that if used properly, and I'm not going to go into the details of what this means properly, it can significantly reduce infection risk. And actually, it can do more at other situations. So, of course, it will take care of outdoor particle pollution, like, for example, during outside smoke from urban pollution or fires. Now, you probably can't see this, but this is a front cover of a WHO document on communication where this aspect is stressed. One point of my personal worry here, with some jurisdictions buying hundreds of purifiers, which are used now, well, in no time at all, once the pandemic is over, they will become potentially electronic junk. But taking this aside, it will work, but of course, it will not do anything to carbon dioxide, VOCs, and any other gaseous pollutants which are generated inside because there's no ventilation. Is this a problem? It's a rhetorical question. We all know that it is. Accumulation of CO2 concentration impacts cognitive function. And there have been many studies on VOCs and other gaseous pollutants emitted indoors and what they do to our health. So air cleaners without ventilation is not a satisfactory solution. Now, I'll come back to the issue of cleaning options in a moment, but I'll now would like to switch to mechanical ventilation. So mechanical ventilation design versus performance. Here we are in a different situation. So there's mechanical ventilation, means there is ventilation. It can potentially be increased enough to reduce infection risk sufficiently. We'll discuss what it means. And potentially it works well in that sense that it takes care of particles of outdoor-indoor origin efficiently by filtering, and then sufficient outdoor air is provided to remove indoor-generated gaseous pollutant. Of course, these are issues in many settings, but let's assume that this is working. So, how much ventilation is really needed for good indoor air quality? Well, it depends where you look or what document you consult. Minimum recommended ventilation rate. All right, WHO, non-residential settings, 10 liters per second. And this is, after this particular document, it's mentioned in that roadmap to ventilation. Now, if you look at the recommendation of ASHRAE, it's a big, long table. And I'm just putting two values, 9.5 and 4 liters per second for an art class and lecture hall with fixed sittings. Well, why it is so much lower for lecture hall? Well, potentially in the art class, pollution is generated from painting, so ventilation needs to be higher. Riva, 10 liters per second per person, according to the WHO. Okay, In two following screens, I have just a shot of how these values are derived, but I'm not going to discuss them here. But the point is that since we are talking about liters per second per person, so how will the building space know the number of people inside and what they do? Are they painting or are they sitting quietly? So that's one issue. And the second is, do we know that these values are enough to adequately lower the risk of infection? For which pathogen, for which variant? I'll talk about this first point first. As I said, these are some background for the setting this guideline and the ASHRAE guideline. Thank you to Jarek and Alex Mikrzewski, who helped putting this main points together. You can check it out. So the building and us, what the building knows about us. This is a slide, a figure from our science publication last year about ventilation option, and I'm just sort of focusing on this demand control ventilation. So demand control means that it's adjusted to what we do. But how many people are actually in the building? More than the design? Well, it's underventilated, less than the design. The design for it's a waste of energy. Building doesn't know how many people are in the building. Of course, there are technologies for this, but in the reality, in practice, it's not something which is commonly used. And the next point is, what do they do in the room? Activities in the room designed for working on the computer or something completely different? Talking or painting, which means emissions of different types. So the building doesn't know what we do, and therefore the building doesn't know how a ventilation we need. But coming back to this second point about infection transmission, I titled this Limits of Ventilation. So what is the necessary amount of ventilation? To find out, we can conduct airborne infection risk assessment. There are models based on Wells-Rayleigh equation, and during the pandemic, extended and much more sophisticated. There were two webinars run by ESIAC that was in November and beginning of December 2020. Last year in Healthy Buildings in June, there was a workshop on this topic. So there are many different models which can be used for this. So an example of one of the model, well, example because this is the model which we've developed, our little team, airborne infection risk calculator. And this is a paper published using this model, and it's that figure which shows how it can be used and what it does. I'm not going to go into details in this, but it is used in that sense that the Italian authorities recommended using this model in their recommendation about infection risk mitigation. So yes, this is possible. There are many different models which were discussed during these events, or the one which was published by Bazant and Bush, which looked at slightly different aspects than the issue of masks, when it's safe and not. So we have models. One point of caution, of course, or limitation is that the assumption here is that there's uniform mixing when using all these models, which is often not the case, but we'll leave this issue aside. So great, we have a solution. We'll have how to control the risk of infection. We use a model, we calculate the required ventilation, and we set up the ventilation, and we are done. Risk of infection is controlled. Is it that easy? Well, not really. Just think about the fact that every venue is different, every setting is different. The model would need to be run for every venue. Will every school, shop, gym, or whatever run the model to set up the ventilation? And then, even if they would, a new pathogens, new variants like new COVID variants would appear, and we will not know the basic parameters to enter into the model, and in particular quanta emission rate. So what do we do? Well, it's not possible then. So this, but then on the top of all of this, there's yet another issue. How much ventilation will actually be needed? So the ability of ventilation to mitigate risk of airborne transmission. And when talking about this, we are talking about individual risk of an exposed person and event reproduction number, the expected number of new infections arising from a single infection individual. All right, so to answer this question and to find out, we did a little study which was published last year. So we considered a typical classroom studied by Wells and a military barracks studied by Coach et al. We calculated ventilation, did calculation for ventilation rates of 2.3, 6.6, and 14 liters per second per person, and we assumed one infectious occupant. Okay, so first of all, the predictive quanta emission rates, what they were. So if we look at this comparison here for a standing and speaking situation, so what do we see? We see, and we did it for a number of different pathogens, as we can see here. So we can see that the values differ. The highest is for measles, the lowest in this diagram is for SARS-CoV-1. SARS-CoV-2 is here, but this is an earlier variant, not Delta and not Omicron. If this was Omicron, it would be much, the value would be much higher. So these are the values, and the methods how it was derived is described in our paper. So what are the risks? This diagram shows it. So we've got event reproduction number on the bottom and individual risk on the top, and we've got these pathogens. Now, the point is that if we look at this part of the diagram here, where the event reproduction number is higher than 1. And for what pathogens it is? Well, it is for these pathogens. And what ventilation it is? So even at the high ventilation risk, ventilation is not going to help to keep the number below 1. And the very same situation is for the calculations for barracks. So ventilation is not really the full solution. So the conclusion from this was that for these viruses, including SARS-CoV-2, even at high ventilation rate of 14 liters per second per person, which is higher than any of these recommendations, it will not be sufficient to maintain event reproduction number below 1 in a fully susceptible population. Well, so these are the limits of ventilation. Is this a new concept? Again, hardly new concept. At Nardell already discussed this quite a long time ago in this 1991 paper. Theoretical limits of protection achievable by building ventilation, limits of ventilation. Well, if we've reached the limits of ventilation, what do we do? And that's why I'm coming to the solutions, which disinfecting the air is one, and perhaps the key one. So what can we do to control the infection of such pathogens for which ventilation is not going to be a full solution? Disinfecting the air in a way that no additional pollution is generated indoors. And I'm stressing this as much as I can. During this pandemic, I have seen potentially every possible solution thought about how to clean the air, and most of them are not solutions because they will generate other pollutants. But the one which seems to be optimal in terms of not doing is GUV air disinfection. Low energy requirements, does not generate new pollutants, is silent, and it's safe. If we are talking particularly of the far UV, like 2020 nanometers, it potentially is the solution. We stressed this aspect in our paper 2 years ago at the beginning of the pandemic, but we focused then was more on ventilation, not on air disinfection. Perhaps we did not fully realize the scale of the problem or the effectiveness of this virus. Application of GUV, again, is this a new concept? By no stretch of imagination, it is a new concept. This is one of the first papers on this topic. So here we are only 76 years behind, and there have been many applications of this and many successful applications. So is disinfecting our air by GUV a solution? Could it be used in every shared space to supplement ventilation? Is it effective, safe, robust? By robust here, we are talking low maintenance, such that it can operate without continuous adjustment and manpower and low cost. Well, it could be doing to the air what we already do to the water. Every drop of water we drink from the tap is disinfected. Every swimming pool is disinfected. Not that we drink water from the swimming pool, it does happen. But the concept that the disinfection is the solution is, again, not new. So there's a lot of work which still need to be done to sort of fully grasp what would be the solution. And something which came across my mind as a possible problem. We are talking about every shared spaces, but homes. Should it be used in homes? And here, my worry is that it shouldn't, because if we use it at homes, we are potentially over-sanitizing, and it would create the problems where we've seen with that over-cleaning. So potentially, no. Well, there's another aspect to all of this, enforceable indoor air quality standards. In September 2021, WHO published this new air quality guidelines. This was immense work which went into this. I was a co-chair of this activity. And if you can see and I'm sure you've seen it already, the values of most of the pollutants went down quite significantly, and basically went down to some of them to background levels like particulates. Because it's not attainable in many places. So there are also interim targets, which could be sort of like benchmarks to gradually, for the jurisdictions, get to the level of WHO guidelines. Now, the point here is that this document is for indoor and outdoor. It stresses in the document that these values apply. These are health-based guidelines, and they apply to indoor and outdoor. Are there any WHO indoor air quality guidelines? Yes, there are. There have been several documents, but one which directly sets concentration levels for specific pollutants is that one for selected pollutants from 2010. So we have the list of pollutants. Some of them are the same as in the outdoor, carbon monoxide and nitrogen dioxide. Well, but we don't see particles on this list. So why not? Because it was stressed that they are the same as the values are the same as the WHO air quality guidelines. Now, the values change from what it was then for carbon monoxide and nitrogen dioxide, but the concept still is the same. Now, this brings an issue and focus on an issue here when we are talking about standards for indoor air quality. It's that interaction with outdoor air quality. So controlling indoor, we cannot avoid linking to the outdoor. And this link is very different for different settings. Some years ago, we've done a summary, some analysis based on published literature data on the impact or the main sources of PM2.5 and PM10 and particle number concentration, we call it ultrafine particles, in homes, schools, offices. So you can see that in homes, for particle number, the indoor sources are the main driver, but outdoor also contribute. For schools, it is the other way around, completely the other way around. Indoor sources are the main ones. For offices, all of them come mainly from outside. So we cannot regulate indoor air without somehow considering outdoor air. So the management of air quality and the issue here, so WHO develops air quality guidelines, and they are used for countries to develop their own standards. And most countries have outdoor air quality standards. And in most countries, there are, to at least to an extent enforceable. But most countries do not have indoor air quality standards. We've just summarized this in a handbook of indoor air quality, which the chapter which I co-chaired for this publication has been accepted. So this is a very depressing situation. There are no indoor air quality standards, and even in the countries which in theory have them, they are not enforced. So indoor air quality is a regulatory no man's land. What is the key issue? Why is this a problem? The main issue and the main obstacle is monitoring. Monitoring is a key element of enforcement. If you will say that the concentrations are to be regulated at a particular level, we must be able to monitor this, and the monitoring procedures must be prescribed. Some years ago, with colleague, we've done this analysis on the application of real-time sensors for indoor air monitoring and challenges to deploying them in urban buildings. We then concluded that the issues are the awareness or lack of the awareness and the lack of availability of appropriate regulation in order to be able to monitor. I guess we were then a bit over optimistic in terms of using the real-time sensors and what they can do. Since then, my team and with colleagues from around the world, we've run a big project on low-cost sensors, and we've applied them for many different solutions, for many different applications. But it is not yet at the level that in every shared space, they could be a sensor or sensors for all these pollutants, which should be monitored for compliance with indoor air standards if they were set for these pollutants for which we have WHO outdoor air quality standard guidelines. So really what we need enforceable indoor air quality standards, and this means concentration levels of selected pollutants, and I'm stressing selected pollutants. We cannot dream that we will be able to monitor all these pollutants in every space. But if they are not measured in every space, if there's no enforcement, this is not going to work. So to finish the vision for the future, and I could say I have a dream. There will be no naturally, in quotation mark, ventilation spaces, in particularly homes that have no ventilation. Indoor air quality standards will prescribe concentration of indoor pollutants, selected pollutants, and be enforceable. Ventilation as part of HVAC system will be an element in enforcement of indoor air quality standards. And ventilation in shared spaces will be supplemented by GUV to control airborne infection risk. A little reflection here. And I couldn't believe when I looked some years and years ago into asbestos issue to find out that it took 2,000 years for the recognition of the asbestos as a risk, a health risk, this which happened during Roman times, for regulations against asbestos to be introduced. 2,000 years. I'm just hoping that it will not take 2,000 years before we have healthy air in all our interiors. Thank you very much for your attention.
Thank you, Lydia. Um, okay, now I'm pondering the 2,000 years. In the interest of time, we've actually got some questions in the chat, but I think what we'll do is we'll go on to our next plenary, and then at the end we'll do questions then, but you have some questions about the survivability of SARS-CoV-2 and aerosols and how that might affect your ventilation. So you can certainly also go into the Q&A and answer directly for everybody. But next up is Richard, who's going to introduce Jordan Peccia for us. So thank you very much. That was fantastic.
Lydia, thank you. Thank you so much. It's wonderful you were able to take the time to present an outstanding presentation. I wanted to very quickly introduce Jordan. Jordan Peccia. Many of you know Jordan, but he's an associate professor of environmental engineering at Yale University. Research and Jordan's lab integrates engineering and public health approaches with quantitative molecular biology tools to study human exposure to microbes in the built environment. His lab is definitely a leader in applying DNA-based technologies for the analysis of indoor air, surface, and dust samples. He serves on the board of directors for AAAR and is an associate editor for the Journal of Indoor Air. It's also my pleasure to be able to work with Jordan in the past so many years on different projects, and we're currently working on one related to mold, normal fungal ecology, etc., which he may make reference to, and we're going to be talking in the bioaerosols workshop immediately after this session. So with that, Jordan, take it from here.
Thank you very much, Richard. Let me just share my slides, and I'll be ready to go. Well, thank you everyone, and it's late afternoon here, so good afternoon, or good morning, or good evening, wherever you may be. So this talk is about measurements in the indoor environment, particularly with reference to dampness, moisture, and mold. When inspectors go into an indoor environment and they do an inspection for a health issue or a mold or moisture damage issue, they often take measurements. But indeed, we don't have great ways to analyze those measurements to tell us if there's a need for remediation, or to tell us if there's a health risk, or even to tell us if remediation has worked and cleared the building. But I think that we can do this. And so what I'd like to talk to you today about is a framework for trying to do this. I can't give you an overall completed test or a tool yet. We're working on it. But I think what I can do and hope to do this afternoon is give you the scientific underpinning for what that might look like. So, you know, if your building is flooded and you have a mold problem as this, you know it. You don't need any fancy measurements. But you can get into a lot of situations where mold is hidden. It can be hidden under floorboards. It can be hidden behind wallboard. It can be hidden behind all kinds of building materials. And then there's situations where there's just mold in a corner, a little bit under a baseboard. Maybe that extends for a larger direction. And the question is that too much? Is it enough? Does it really impact things? Do I really have any exposure to that mold? Do I need to worry about it? Indeed, in a previous study from Lawrence Berkeley Labs, they estimated nearly 50% of homes in the United States at least self-report some type of dampness or mold damage. And this isn't getting any better. Certainly, as climate continues to change, we are having these extreme precipitation events more and more frequently. And we're certainly having them in the northeast and eastern part of the United States. And as we get that, it's associated with flooding, sea level rises, and all kinds of issues with moistures in buildings. So I think it goes without saying that mold is bad for you. But, we only know that and only have built associations of mold being bad for you or you having some sort of respiratory or health risk if you can see it or if you can smell it. And it seems like we can do better than that because we often hypothesize or at least make this direct link that it's the mold that is causing the problem. Although the measurements that we try to make with mold, which I'm showing down here, do not really result in showing high odds ratios. Rather, it is the moisture damage, the visible mold, or the mold odor that has the strongest associations with those health effects. This is a sort of a meta-analysis put together by Mark Mandel and Rachel Adams in 2019. But the mold should work, and my theory here is that we're just not taking the right measurements. We don't have the appropriate ways to think about this. So here's the setup. Water damage, negative health impacts, visible mold, negative health impacts, odor as well. But can we do this connection? Can we say that fungal communities are meaningful here? And is a fungal community, this is the basic question, in a damp building or a moldy building, even if the mold is hidden, is it fundamentally different than that of a dry building or what we might want to call a normal building? So here are the experiments, and I would say that it's not as an obvious question as you may think. Again, if it's a heavily flooded building and you have a lot of visible mold, that's more obvious. But there are many, many different sources of fungi mold inside of a building. They're brought in with ventilation. There's large amounts of fungi in outdoor air that get swept in, that get tracked in. Our dogs, our babies, everybody has some sort of fungi food as well. The difference between a moldy home and a not moldy home might be a small amount of growth in the corner, underneath a baseboard, etc. And so you can see, I think, from these graphics that trying to pull out that signal might be difficult. So in order to answer that question, and this was work that we did with Richard Shaughnessy and Ulla Havarnan Shaughnessy at Tulsa University, we did a national study where we enlisted and had great participation from several remediators all across the United States. They looked at homes that were moldy and then also sampled homes that they knew, or to the best of anyone's knowledge, had no previous history of mold damage. We found mold homes in both categories. We collected settled dust from the tops of door frames in each of these categories. And we did so indoor, outdoor. We even looked at the direct mold as well. So all of this together results in hundreds and hundreds of samples. When we do our identifications, it results in millions and millions of identifications of different mold species. And so if you put it all together and you start asking that question, is the air in a moldy home different than the air in a home that has no history of water damage or mold? The answer is yes, but the differences are subtle. So you're looking at here on the y-axis what we call observed ASV count. Every time you see ASV in here, just replace it for species, if that makes it a little better for you. We're trying to be technically correct here and use the term amplicon sequence variance because we're not really identifying species. But what we're looking at here with each dot is a number of species within a sample. So direct mold, the species are depressed, obviously, because there's just one or two different taxa that typically are directly on a moldy material. But the air in a moldy home is also depressed compared to the air in a not moldy home. And it's also different than the air outside. We don't find any statistical difference between the diversity or the richness in a moldy home or in a not moldy home versus the outdoor air, but we certainly see a statistical difference when there's mold present in that home. We can make some connections there because we had previously shown, and certainly several other authors had shown this, that when fungal diversity is depressed, especially when children are exposed to that early on in life, they have a greater chance of developing asthma. Here, the odds ratios were large. Our work here is with collaborators in the Chamacos cohort. This is another American cohort. These are two European cohorts that saw the same thing. Asthma development was inversely related to diversity in mold. And we, of course, see this in a home right now. That is that a mold damage in a home tends to depress the diversity in that home. But there's a lot of other signal as well. Let's compare now, again, this is number of species in common. Let's compare the outside air to the air of a moldy home and the air of a not moldy home. And what you can see is There are fewer things in common if it's a moldy home. There are more things in common if it's a not moldy home. That is, a not moldy home looks like outdoor air. A moldy home is different. Keep on going. Let's now compare direct mold, the species and the communities that are growing on moldy materials, to the air in a moldy home and to the air in outside. And again, you can see that there's not a lot in common between direct mold on a material in the air outside. There is more common between direct mold material and the air in a moldy home. More signal. Why don't we just put all these homes together, put all these samples together, line up the species and say, in a home that we have evidence that there's mold, there's visible mold or moisture, versus a home that we have no evidence that there was ever a mold or moisture problem, what are the differences in the taxa and which taxa really stand out? Well, these taxa of Aspergillus, Alternaria, Penicillium, Stachybotrys stand out in the moldy home in a statistical relevant way. Again, I'm just giving you the genus here and the name of the sequence variant. We can run these and we can get an exact sequence for them, but sometimes there's the taxonomy is not completely clear. So we just prefer to call these an ASV number versus identifying, the closest match with the species. You can do the same thing, and you can say, how is a moldy home different than a not moldy home? And when we put all these homes together, of course, there's lots of overlap in them, and there should be, but we see things like yeast from humans, Malassezia, is more dominant in these not moldy homes. So there's a lot of signal here, but it's a complex signal. It's not just one species that we're going to compare from one home versus another home. So we need a way to handle that complexity. And so we move towards machine learning, and in this case, random forest. So random forest is just building a decision tree. It's looking at a sample and it's saying, is there a high Aspergillus concentration? Yes or no? No. Then what about a high Malassezia concentration? Yes or no? And it's moving down and trying to make a decision whether this is a moldy home or not moldy home based on different types of training sets from what a moldy home and a not moldy home looks like that. We can run these individual decision trees on a sample, and then we can run many, 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 many different decision trees on a sample, thus called a random forest. And we can even run many different random forests. In this case, we do 20 different random forests per sample, and we get predictions that look something like this. That is, in a moldy home, about 45% of the time, our predictions are correct. That is, 20 out of 20, we predict that it's a moldy home or project that it's a not moldy home. These are still good projections. 15, 16, 17 times out of 20, we're correctly predicting it, but we have some stuff here on the other side of 10. This is 50-50. We don't really know it's one or the other. This is, we're decidedly incorrectly classifying that home. If you put all that stuff together, we can tell a home is moldy or not moldy just based on the microbial ecology in a random forest model about 75 to 80% of the time. But if we look at all the samples in the home, that is, if we pool them and say there's 3 samples in the home exactly, if 3 out of 3, then yeah, that's a moldy home. If it's 2 out of the 3, we're still going to call it a moldy home. And the same thing, if it's not a moldy home and it's 2 out of the 3 are right, we're going to call it a not moldy home. When we do that, we can move up to 95% correct classification. So a way to look at this, and let's just go down through the moldy homes, is that we need 2 out of the 3 samples correctly classified in order to do this properly. And you can see in the moldy homes, everything is correctly classified here, because over there in the incorrect classifications for samples, there's never more than 1. The same thing with the no moldy homes. The only case where we incorrectly classified was a home in Minneapolis, Minnesota, that tended to have a low fungal diversity, even though it was not a moldy home. So the answer to that question is, what about fungal communities in indoor air? Are they distinct between homes with mold and no mold? I think that they are. And I think that with DNA-based community profiling and adding in this complexity of running a machine learning algorithm that's trained, we can do that and we can ferret out what these differences are. And with at least so far 95% accuracy, we can predict whether that's going to be a moldy or not moldy home. Now, so that's the answer to that first question. But I have a second question here, and this gets a little bit more towards the health. There's a lot of things that we could do with the health here, but I'm going to talk specifically about, is there another feature here? Is there another thing at play about fungi that makes us sick? And so that's our second question here, and I want to get back to this graph. And this graph has sort of a clue to it. That is, what about visible mold or water damage or mold odor so strongly correlates with health effects when the actual raw measurements of a spore doesn't correlate with those health effects? So this is sort of telling us, maybe even if we do measure correctly, it's telling us who's there is important. It's what the odor is, or what the visible, how much moisture there is in the room. This makes a bit of sense to me because, several years ago, we did these same type of experiments to just ask the question, in terms of, say, an allergenic response in a human, is a Aspergillus fumigatus spore the same all the time? And the answer is no. You may count exposure to 1,000 Aspergillus fumigatus spores, but if those spores were grown at significantly different temperatures for sporulation, there can be orders of magnitude differences in the health effects. So again, you're exposed to 1,000 spores, but if it's grown at a low temperature, it's going to be much more allergenic than if it was grown at a higher temperature. Again, that makes sense because the proteins that are allergenic are coded by genes, and gene expression, of course, can be inducible, and it's inducible by all kinds of environmental factors. It only makes sense that these environmental factors that can induce some of these allergenic genes also correlate to the health effects. So in some work that was done by Karen Dannemiller, who's now at Ohio State, but when she was previously in my lab, and Bridget Hegarty, who is now at the University of Michigan, we tried to assemble a metatranscriptome. And that is not looking at just one gene, but let's look at all genes. And the environmental factor we care about here is just going to be moisture, that is moisture content of a material. And so in chamber setup with house dust from Karen's mother-in-law, that's why it's pink. That's why it was thrown away and available. We set up chambers at different water activities. We allowed for time microbial activity to happen. The water activities were 0.5, 0.85, and 1. We extracted the RNA out of that house dust. We sequenced it and sequenced it and sequenced it and got to the point where we could assemble all that together and look at gene expression, what was going on genetically in those samples. And so here's what's going on. This is going to toggle back and forth. And so if I can direct you up to the upper left-hand corner, you can see the water activity changing. It is changing from 0.5 to 0.85 to 1. And when it changes, you can see parts of this metabolic cycle. This is called a KEGG diagram. It's all the metabolic cycles of life. And when it lights up versus when it's in the background shade, that lit up stuff, these are metabolisms that we are actively seeing going on in a spore. At 0.5, it's low. It increases at 0.85, and it really lights up at 1. That is, secondary metabolites are really going at 0.85, and especially at 1. At 0.5, there's not a lot of activity. It is maintaining, it's staying alive, it's staying a spore, but it's not producing a lot of odors, a lot of VOCs. It's not running a lot of secondary metabolisms. If we go through all of those genes, we pull out the ones that we know are to be allergenic genes. And then we look at the different expression of them under the different conditions, we can get graphs that look a little bit like this. And just to kind of explain this graph, what I'm comparing here is the water activity of 1 versus the water activity of 0.5, and whether it is overexpressed at 1 or overexpressed at 0.5. That's a way that you could look at that. So again, more highly expressed, I'm sorry, I had this opposite on this graph at 0.5, and this is more highly expressed water activity of 0.5 than 1. And what we see when we go through these is that we typically have more expression when the water activity is higher. It's hard always to tell these, but you can see the numbers where things are more highly expressed on top. We see the same thing with mycotoxin genes, higher expression at higher water activity. We see the same thing with secondary metabolites and VOCs as well. And that leads us to believe that, again, a spore is not a spore. Even if you have the same microbial ecology in one building versus another building. If there is moisture, more moisture in one building, and there is more metabolic activity, that ecology could be producing more genes that are allergic to the occupants of that building, and you could have a different health respect. That is, you could have a different dose response curve. We shouldn't be thinking about dose response curves in terms of this many species, this number of this many species has this a health effect. We have to be thinking about what are the conditions in which that's growing in. And that makes sense, some sense when we go back and we look at these final graphs. Let's see, did I have a final graph here? Nope. When we look at that final graph, sorry about that. When we go back, we looked at the final graph of the Mendel and the Rachel Adams paper that showed that at a higher moisture content, we see greater health effects. We also see greater health effects when you can smell the mold, when the odor is detected. Those are conditions where things are wet versus low water activity when no odor is around, but there was still perhaps some mold damage and lingering fungi around. Those are conditions where things were dry. We're hypothesizing at least that conditions when things are dry is a significantly lower health risk than it is when conditions are wet. There's a lot more work to be done, but I think that if we pair the ecology together with the microbial activity, we can get closer and closer to coming up with tools to be able to solve these problems. Now, some of these tools may look a long way off, and you may say, well, we can't all go down and start a fungal metagenome at the point of inspecting a house. We all can't do sequencing. But the truth is, the sequencing now is pretty inexpensive. You can sequence a sample for less than $30. That sequencing is becoming more and more available. Turnaround times are very quick. And the process of reverse transcriptase and quantitative qPCR, especially after COVID, because this is how COVID was tested, has become so rapid and so automated that these types of analyses are not far off in the future of being quick, rapid, and low cost. We just need to know a little bit more on what to look for, and we need to take all of this type of data and we need to ground truth it in epidemiology studies to make sure that what we hypothesize our health effects are there, and also whether we can be used in clearance and in remediation. So with that, I think I've left a few minutes in here for questions from the chat. I just want to acknowledge some of the key players in here, the funding from HUD, the National Science Foundation, the Sloan Foundation as well, Bridget Hegarty and Karen Dannemiller, Ulla Harvarnanen, and Richard Shaughnessy. Thanks. I think I'll end there.
Excellent. Richard, are you there? Yes. I was going to at least ask one of the questions from the Q&A. Is that okay?
Yeah, I saw one too. I'd like to
Go for it then.
to address. Had to do with Jordan, and particularly it has to do with what you're talking about in terms of moldy and no moldy homes. And the question is, since visible mold, odor, and water damage are sexual liable predictors, aren't there aren't these still more useful to trigger remediation? Admittedly, they are subjective, but there could be quality problems from broad application of testing, which goes to this kind of Jordan, I think, and there's another question very similar. It talks mold or no mold homes and classification tools. How would you respond to that?
I think there's a few things to say there. The first thing is this is an academic study, so when we wanted to see if there was a difference in the microbial ecology between a moldy home and a non-moldy home, we could not leave any ambiguity in whether that was a moldy home or not. That is, we're not going out and trying to test in a home that, say, somebody has a complaint of whether that's a moldy home or not. We have to do this on homes that have visible mold, because we have to know that it's a moldy home. So that's the first thing. That's a factor of the study. That's not necessarily how it would always be applied, but that's how the study has to work, because we can't have ambiguity in our control and test cases. But the second thing is yeah, absolutely. They are reliable indicators. If a home has got a lot of visible mold and odor, I mean, you don't need this test. You don't need an inspector either, right? Just get it remediated. But I think many people would argue that maybe you do need an inspector, because maybe there's something behind that visible mold, and maybe you need to dig around a little bit more and find the extent of the mold as well. But even in that case, I would say that once that home has been remediated, it would be lovely to have a test that you could say, yeah, this home is cleared. This home now looks like a normal home. It would also be great in the many, many cases when inspectors are called to a home and people have a health impact, they're not feeling well and they suspect mold, but perhaps they can't find mold, to at least give a definitive test and say, look, in 95% of the cases, this test indicates mold. It's not indicating mold in your home. And so, that's at least something that you can tell them. Versus saying, I can't find anything, but maybe there's something there. So again, I think that test that is definitive, quantitative, and backed up with statistics can be a useful tool.
Thanks, Gordon. Carrie?
I think that we've had two fantastic talks, but I think we're actually at the limit here. We're getting ready for our next session. So I wish we were in person, we could hear the applause from the audience, but you'll just have to take my word for it, they're applauding. And so thank you very much. And also, you will see Lydia was able to answer some of the questions that were posted, and Jordan, you will also see that you were given some to directly answer as well. We're getting ready for the next Ready Room, which will happen here in a moment. But thank you very much. And again, if there are questions, certainly, hopefully folks can get a chance to reach out to you directly as well.
Thanks.
Thank you.
Thank you all.
Thank you.
Thanks, Lydia. Thanks, Gordon.
Thanks. Bye.
So much.
If you're not attending the next session on this stage, please exit out of WebEx by clicking the red X button at the bottom of your screen. Thank you.
Thanks, Lydia.
Thank you.